years, in my last lecture, I have discussed with you the timeline which led to the discovery of the structure of DNA. Not exactly discovery, but the elucidation of the structure of DNA. Then I also discussed with you that DNA is, the nucleotides are made up of three components, that is sugar, a phosphate group, and a nitrogenous base. I have discussed with you that DNA has deoxyribose sugar in it, whereas RNA has ribose sugar in it. Then we went on to discuss the nitrogenous bases, and I told you that the nitrogenous bases are of two types, that is, they are purines and pyrimidines. Then we went on to discuss in detail the structure of pyrimidine, and we found that pyrimidine molecule is very similar to that of benzene. That is, pyrimidine molecule is made up of 4 carbon atoms and 2 nitrogen atoms which are present in a ring form, and these carbon and nitrogen atoms are connected via alternating double and single bonds. So when they are connected via alternating double and single bonds, this structure allows for the resonance or aromaticity, causing the ring to be very stable. So as you can see on your screens now, the 3-dimensional structure of a pyridine molecule. And what do we find? That now, this I've already discussed with you, that these atoms can be numbered starting from the bottom nitrogen. So you will always give number 1 to the nitrogen which is at the bottom, and then we will continue to number it in such a manner so that this nitrogen will get number 1, this will get 2, this nitrogen will get number 3. Supposing we had drawn the structure in such a manner that we had interchanged this nitrogen with carbon, so we would have numbered it in 1, 2, and if nitrogen was here, this would have been 3. So irrespective of where the nitrogen is placed, because these two positions are equivalent, so we will say that we will number it so that these nitrogens get the lowest possible number. So as I was said, by convention the atoms are numbered so that the nitrogen atom gets the lowest number. So N1 and this N1, this nitrogen which is getting number 1, this is where it is attached to the ribose sugar in case of RNA, and it will be attached to a deoxyribose sugar in case of a DNA. So this is the basic structure of the pyrimidine ring, and you've seen how we've numbered it: 1, 2, 3, 4, 5, 6. Now what you have to remember is that this is the basic structure of a pyrimidine molecule. There are many derivatives of this structure, and through the addition of one or more functional groups. So what we mean is that if we replace one of the carbons or we attach one of the groups at the carbons, we will get derivatives of these molecules. So when we talk of pyrimidine bases, so what do we say? The pyrimidine nitrogenous bases are derived from organic compound pyrimidine through the addition of various functional groups. So when we are talking of pyrimidine nitrogenous bases, we are talking of the substitution on the basic pyrimidine molecule. The term pyrimidine is also used to refer to pyrimidine derivatives. Most notably, remember the 3 nitrogenous bases: thymine, cytosine, and uracil. So these are the 3 pyrimidine bases. Out of these 3 pyrimidine bases, thymine is only present in DNA. Uracil is only found in RNA. and cytosine is found both in DNA and RNA. So how do DNA and RNA differ? You have to remember, in DNA the sugar is deoxyribose sugar, whereas in RNA the sugar is ribose. Then they differ in their pyrimidine bases. In DNA we have thymine, whereas in RNA instead of thymine we have uracil. Now these three pyrimidines along with two purines, adenine and guanine, are the building blocks of both deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. These derivatives, you have to remember that although we are placing substituents on the pyrimidine molecule and the basic purine molecule, these derivatives will retain the simple 6-membered ring. But the modifications can range from an addition of a few atoms in nucleic acids to complex structures in drugs and vitamins. The three pyrimidine nitrogenous bases, thymine, which is referred by abbreviation is T, cytosine, C, and uracil U, are modified forms of aromatic compound pyrimidine. They consist of a 6-membered ring with 2 nitrogen atoms and 4 carbon atoms. So this is the basic pyrimidine ring which will be present in all nitrogenous bases. So instead of being an aromatic ring now with alternating double and single bonds, they all have a ketone, that is a carbonyl group, on the second carbon atom, that is the carbon which is between the 2 nitrogen atoms. And you know, when we will add the carbonyl group to the ring, it will remove the double bond from the ring. So it will remove one double bond, and this in turn will result in 2 double bonds and 4 single bonds. So what you have to remember is, whether we are talking of thymine, uracil or cytosine, what we'll find is we will be replacing one double bond with a carbonyl group in all the three. And this will result in what? That instead of having 3 double bonds now in the molecule, we will have 2 double bonds and 4 single bonds in the molecule. And together with the carbonyl group, these pyrimidines may also have some additional functional groups. So when we talk of cytosine, what do we find? That cytosine is a modified form of pyrimidine, where in addition to the ketonic group present at C2, there is an amino group present at C1, at C4. Hence we name it as 4-amino 2-oxypyrimidine. So if you look at the structure of the molecule, what do we find? That this is your carbon number 1. At carbon number 2, the double bond which was between this nitrogen and this carbon has been replaced with a carbonyl group here. And then at 4th carbon, we have a amino group. So instead of having 3 double bonds here, now we are having 2 double bonds and 4 single bonds. And along with that, what do we have? We have a carbonyl group at the 2nd carbon and the amino group at the 4th carbon. So this is the structure of cytosine, and this is named as 4-amino. We are having an amino group at 4th carbon, pyrimidine, 2-one. So what do we mean by 2-one? That is, at the 2nd position, we have replaced the hydrogen with a carbonyl group here. So now go on to the next structure. So if we talk of uracil, uracil has 2 carbonyl groups. So what do we found that in cytosine we had 1 carbonyl group at C2, and this CO2 group, the carbonyl group at C2, is common to all the 3 pyrimidines. The second structure modification or the functional group which we find in uracil is, that is, we have one carbonyl group at C2 and the second carbonyl group is at C4. Hence, the name would be 2,4. That is, we've replaced the hydrogens here with oxygen. So we have 2,4, 2,4 dioxypyrimidine. And if we talk of the common name, the IUPAC name, it would be pyrimidine 2,4. That is, at the second and the fourth position, we have ketonic group. So we are taking the suffix, that is, di indicates 2 and on indicates that it has 2 carbonyl groups in it. Now we go on to thymine. And when we talk of thymine, we find that in thymine, a methyl group is present at C5 along with the 2 carbonyl groups at C2 and C4. So in uracil, we have 2 carbonyl groups at C2 and C4, whereas in thymine, we have a methyl group present at 5th carbon along with the 2 carbonyl groups at C2 and C4. So the name will become 4-methyl 2,4-dioxypyrimidine. So this, you can also see the structure of this molecule on your screens. That is, we are having 2,4. That is, at second and the fourth positions, we have the ketonic groups, and at the fifth position, we have a methyl group. So we name it as 5-methyl, we take this as a substituent, pyrimidine, which is your basic molecule, and 2,4-dione, that is, the carbonyl group is present at the second and the fourth carbon. So that means the addition of another ketonic group in thymine and uracil removes another double bond from the ring. So when we are adding another carbonyl group, we are removing another double bond from the ring, having only one double bond in uracil and thymine and 2 double bonds in cytosine. So now if you recollect the structure, what we know now is that cytosine has only one carbonyl group. So since it is having one carbonyl group, it will have 2 double bonds. Whereas thymine and uracil has 2 carbonyl groups in it. So since it is having 2 carbonyl groups in it, these will have only one double bond in them. So here you can see the structures, that when we look into the structure of cytosine, we have only one carbonyl group here. So since we are having only one carbonyl group here, what do we find? We have a double bond here, and we have cytosine, what we find, we have, this is the structure of cytosine, that is, this is the structure of uracil, that is, we are having only one carbonyl group here, and since we are having only one carbonyl group here, what do we find in this structure? We have only one carbonyl group, so we have 2 double bonds. Whereas in thymine, what we find, that we have 2 carbonyl groups, and since we have 2 carbonyl groups, we are having only one double bond. Similarly, we find in this structure that this structure is also having 2 carbonyl groups in it, so it is having only one double bond in it. So now these are the 3 bases, that is cytosine, thymine and uracil, which are present in nucleic acids. In DNA, we have thymine and cytosine, whereas in RNA, we have thymine and we have cytosine and uracil. So now let us go on to discuss the structure of purine. This is the second base which will be present in DNA and RNA, that is the derivative of purine. The word purine, that is pure urine, was coined by German chemist Emil Fischer in 1884. He synthesized purine for the first time in 1898 by a reaction sequence starting from the uric acid. Earlier uric acid had been isolated from kidney stones by Carl Wilhelm Scheele in 1776. And we know that purines are heterocyclic water-soluble aromatic compound with a chemical structure made up of imidazole ring fused to a pyrimidine ring. Purine is a crystalline base with the chemical formula C5H4N4. It is the parent of a compound of uric acid groups. Purines include substituted purines and their tautomers, and they are the most widely occurring nitrogen containing heterocycles in nature. Purines A and G make up nucleic acids, that is, adenine and guanine are present in nucleic acids, DNA and RNA, and they are also present in certain alkaloids such as caffeine and theophylline. Purine has 2 carbon rings and a total of 4 nitrogen atoms. The pyrimidine ring of purines contains 2 nitrogen atoms that are located at positions 1 and 3 of the ring, which is similar to the structure of pyrimidines. The imidazole ring attached to the pyrimidine ring has 2 nitrogen atoms that are located at position 7 and 9. So that means now nitrogens are present at position 1, 3, 7 and 9. The numbering is by convention counterclockwise in the larger ring. However, numbering is clockwise in the smaller imidazole ring. So if you look at the structure of purine here, so this is the pyrimidine ring and this is the imidazole ring. So first of all, you have to remember that you will start the numbering from the larger ring, from the pyrimidine ring. Number 1 will always be given to nitrogen. And then you have to remember that this numbering has to be done anti-clockwise. So whereas in pyrimidine we were saying we can do the numbering clockwise or anti-clockwise when we are looking only at the pyrimidine molecule in isolation, where we shall obey the lowest sum rule. But in this case, this is a fused system made up of 2 rings, pyrimidine ring and the imidazole ring. So you have to remember here that here the numbering will always be anti-clockwise in the pyrimidine ring, and we will start from this nitrogen. So this will be 1, 2, 3, 4, 5 and 6. From here, now when we go on to numbering in the imidazole ring, what we find here that the numbering will be clockwise. 7, 8 and 9. About this numbering you have to be very careful, because why have you to be careful? That in pyrimidine ring, when we are talking of pyrimidines, it is this nitrogen which is given 1, and it is from here it is attached to the sugar molecule. Whereas when we talk of pyrimidines, it is attached to the sugar molecule from N9. So you have to be very careful when you are carrying out the numbering. So this is your N9. In this case, the numbering has to be anti-clockwise in the pyrimidine ring or in the larger ring, and in the smaller ring the numbering has to be clockwise. Now let us look into the modifications which are done in this basic purine molecule. So what we find in adenine, the amino group is present at the carbon 6. So this is the basic structure molecule of the purine. So what we've done in case of purine, a amino group is present at this carbon. So while you're doing the numbering, please remember 1, 2, 3, 4, 5, 6. So this amino group is present at 6 amino, and we call it 6-amino purine. This is the IUPAC name of this molecule. Otherwise, this is popularly known as adenine. So when we'll be discussing the structures, we'll be talking as adenine, thymine, guanine, cytosine, uracil. We will be not talking in the terms of the IUPAC names. Now when we go on to guanine, in guanine oxygen is present at 6th carbon at this position in place of amino group. Please remember, in thymine what was present here? Amino group was present here, whereas in guanine oxygen is present here. So, and in this case amino group is present at 2nd position. So the name, now you have to remember, when we are naming the substituents, we name them in alphabetical order. So the naming would be 2-amino 6-oxy. Oxy group is present at the 6th carbon, purine. So this will be the IUPAC name of guanine. You are doing the numbering 1, 2, 3, 4, 5, 6. Here we have ignored the priority given to the principal functional group. Here the principal functional group is carbonyl group. If we compare the carbonyl group and the amino group, here the numbering is being done according to the ring. That is, in this ring the numbering has to be done anti-clockwise. Please remember this rule: 1, 2, 3, 4, 5, 6, 7. This is anti-clockwise. 7, 8, 9, which is your clockwise numbering. Now what we find that these pyrimidines and purines will exhibit tautomerism. That is, what do we know? What is tautomerism now? Tautomerism is a very important type of isomerism. It is a structural isomerism in which the compounds can exist in two structural isomeric forms which are mutually interconvertible and exist in dynamic equilibrium, and these are called as tautomers. The term tautomer, that means auto, same, meso parts, that is, it is made up of same parts, was first coined by Lahr in 1885. The tautomer containing the carbonyl group is designated as the keto or the lactam form, and the other one having a hydroxy group, or and that is a OH attached to a doubly bonded carbon, is referred to as the enol or the lactam form. This kind of tautomerism is called ketoenol or lactam tautomerism. So on your screens now you can see the keto form. So when you look at the keto form, what do we find in the keto form of the structure of the molecule? We have carbonyl groups. And then what we find, that there is migration of hydrogen from nitrogen to oxygen, and there is a double bond between this nitrogen and this carbon. So this form is known as the enol form. So what is the keto form or the lactam? Where we have a carbonyl group, and the enol form or the lactam form is the one where we have an OH attached to a double bond. So on your screens again I have shown you the tautomerism shown by different purines and pyrimidines. But what you have to remember is that at pH 7 it is the keto form which is more predominant. So when we talk of adenine, what we find, adenine is also present in its keto form, and when we talk of guanine, it is also present in its keto form. So in this till now, what I have done is I have discussed the structure of nitrogenous bases, which are components of nucleic acids. That is, I have discussed the structure of pyrimidines and purines, that is the basic structure, and then I went on to discuss the structural derivatives of pyrimidines and purines, that is cytosine, uracil, thymine, which are pyrimidines, and adenine and guanine, which are purines. Now in my next lecture, we shall be discussing the structure of nucleosides and nucleotides. For this lecture again, I had referred to the books by Nelson David, Cox, Michael Lehinger. Then that is the Principles of Biochemistry by Benjamin Pierce, Benjamin, Genetics, a Conceptual Approach, and by Klug, Williams and Cummings. Then also a book by Garrett, Reginald and Charles. That is Book of Biochemistry. Thank you very much.